Okay, so this is my website. I'm focusing on Fragile X syndrome. And for my introduction page, I pretty much just gave an overview of what the disorder is. And I included this diagram here that shows the difference between a normal and a mutated chromosome. And then I also talked about the causes, how it is transmitted, and then also how common it is in the United States population. And I have this pie chart here that talks about the types of mutations and how common they are in people with a family history of Fragile X and people without a history. And so for my symptoms and signs page, I divided it up into 3 different categories. We have behavioral and social challenges, intellectual disabilities, and physical characteristics. And then this image here shows patients with Fragile X so that the reader can sort of understand the physical phenotypes associated with it. And then I also talked about the diagnosis process. You can do prenatal testing before the baby is born, and then diagnosis of the child, usually when they're around toddler age. And I chose to include this diagram that shows how PCR works, because I did talk about PCR in the diagnosis of children section, and I wanted the reader to have a better understanding of what I was talking about there. For the tissues page, I focus mainly on 2 types of tissues, but Fragile X does impact a wide variety. So I talked about the neurons and the ways that Fragile X impairs brain development and the neurons in the brain, which in turn affects cell signaling between the body and the brain. And then I also talked about the connective tissue because a lot of the physical features that are associated with Fragile X have to do with the connective tissue. And so this image here just shows the different types of cells within the extracellular matrix. For gene signaling and pathways, I gave an overview of what the actual mutation is. And I think this diagram does a really good job of showing and comparing the difference between a normal FMR1 gene and a mutated gene, and how the mutated gene does not result in any mRNA or protein created. And then in this section, I went into more detail about the actual pathways that are involved with the protein itself. And this diagram also does a really good job of showing the difference between a normal, a fully mutated, and a pre-mutated FMR1 gene, and the ways in which the protein impacts the development of the neurons. For treatment, there is basically therapy and medicine that can be used for Fragile X. So I broke down the therapy into the 3 most common types. And then I also included the 4 most common symptoms that are treated for Fragile X. And I talked about how these medicines work and some of the common brands that are associated with them. For research, each one of these paragraphs talks about a different type of research, a different research project that is still being conducted. So this first one talks about the effects of pre-mutations on carriers of Fragile X. The second one talks about a possible treatment that involves the reactivation of the FMR1 gene. And I found this one really interesting because it uses CRISPR, and I included this diagram that gives the reader a breakdown of how CRISPR works, because this is such an important and tool that is being used in genetics right now. So I really wanted to include that on this page. And then this last paragraph here talks about animal models with mice and fruit flies. And then lastly, we have the research paper that I read. I pretty much just summarized the whole paper and talked about the different methods and conclusions that they came to. And then this last diagram here talks about, or it shows the results of one of the case studies that they focused on. So here are my references. I hope you enjoyed my website, and thank you for listening.